Okay, well, we've all come so far. We find ourselves nearing the end of the semester, so it is time for those dreaded group projects. Yeah. The group project, essentially what I am looking for is, the pipeline we've been working on to assemble, annotate, and count all these reads. We've done it for one sample. But it's been done in for loops, so we can easily extend it by just adding accessions to that file of accessions, rerunning it. So great, we could go ahead and do 10 samples. That way we can actually start comparing some of the output. But I'm also really interested in seeing how some of the parameters that we pick affect the conclusions that we would draw. And so the idea here, big picture, is we're going to pick 10 samples for each group. And everybody in the group is going to run those same 10 samples through the whole pipeline, but with at least one parameter change. Now it's up to you and your God to decide what you want to change. Whether it's the fast QC, right? Maybe you decide we're gonna not cut out things that have ends and whatever. Maybe it's in your spade step. Say we're gonna try a different type of assembly where we use larger gamers. 27, 55, and 127. But everybody in the group is going to kind of take the same pipeline and tweak one little part of it. And so we're going to have, for each group, the same 10 samples run through more or less the same pipeline, but slightly differently. And then out at the end, we're going to all have our little files that have we found this gene in this abundance in that sample. So what I'm wanting, like the output from this group project is that your group will give me 30 output files. 10 things, same. Every person owes me 10 output files. Let's put it that way. And I can load those up into R. I've got a little script already that will just like look through and see what genes were present in what abundance. We can compare those across samples. So I've got 10. We want to pick samples that it's like 5 from one treatment, 5 from another. So we can do some stats at the end, like, okay, do people taking this drug actually have different genes being expressed in their microbiome? Does this invasive plant change the functional profile of the soil microbes? Doesn't have to be microbes, that's just always right. So what I want to do today is not get bogged down in the code that isn't quite running correctly yet, but it will by the end of the day. Thanks for noticing that. I'll get that fixed. I don't want to get bogged down in that because that's just like a little technical turd. That's not important. What's more important is that we've got a set of skills that we can actually start using to do biology with, answering questions. I know it's taken us a long time to get there, but here we are. And so now we can put together a pipeline. The whole point of the pipeline isn't to talk to a supercomputer. That's the point of this class, but that's just like you are now tall enough to ride the ride. So now it's time to actually go in and ask some questions. So what I want you to do is get on SRA, the sequence read archive, and we're going to try to find some interesting samples to work with. We're gonna try to find them from the same study, but in the different treatments, with or without that drug, with or without the invasive plant, whatever. The other way to go about this is to find a paper that you like that uses shotgun metagenomic sequencing. We can, that way we know like the background of the samples. So does anyone have just really big picture, an idea of like a biological question that's interesting that somebody may have used shotgun sequencing to address?
So one is like, I know like when this is like there's like the whole like the brain analysis with really different Parkinson's. And so I was wondering if there's like any controls versus patients with Parkinson's that they really that provide.
Okay. So I'm gonna search for Parkinson and metagenome. That's anticlimactic.
No, I did a plus that said the plus was ignored or something.
Okay, so there's a starting point. How can we kind of whittle this down to what we might be looking for? We don't want ion torrent. That's the crappiest sequencing technology of all time. We want Illumina. That's what our pipeline's made for. Mouse 5 doesn't look promising. Right over here. So we can filter by taxon. So human gut. And notice how it's filling out our search. Yeah, ideally paired.
I'm reading.
Because our pipeline is written prepared at this point. I don't want to rewrite it. I don't want to make you rewrite it. At the Thanksgiving dinner table, you can see you there with your laptops, like spilling gravy on them when you try to rewrite this to make it work with single end reads. Got FastQ's Illumina paired for human gut metagenome. And I don't like Parkinson metagenome being a whole search term, so I'm just going to say Parkinson. And well, what have we got? What's 16S? That's not shotgun. All right, this is they amplified the 16S region first and then just sequence that. Our pipeline is for actual metagenomes, where it's all the genes, and that's what we're looking for, is like all the genes that are present. So we don't want any of those. How can we get rid of them? That can help. Genome. We could try shotgun, or wait, where? Oh, here. That's better. I'm gonna send all of those. There's only 154 to the run selector. This will let us look at the library selection strategy. That's like okay.
Could you show like the button, for, like the run select?
Oh,
Is that like the only time like it shows up? Like, remember when I was doing the lab for the assignment we were trying to find stuff on the run selector? Sometimes like that wouldn't show up.
if you didn't have any hits at all.
Okay.
But it's been there this whole time, like once you kind of have your search results, if there are some hits, similar response like work. These look promising to me. Multi-omics of human gut, Parkinson's disease, I'm guessing. There's a lot of samples for that. So go to room selector. Okay. I'm just going to download this whole metadata. Open it in Excel. Just let it out. Okay, we've got the run. That's the accessions that we all know and hate at this point. The number of bases. The bio project, that's basically all the same study that we're seeing here. Those are all the same, but this one's different. Okay. Where it comes from, all this good stuff. Okay, the library selection. This can be a few things. Random versus PCR. So what we're looking for. We don't want PCR because that's that 16S or ITS. So we do want random. Library source, metagenomic, all the way down. Okay, this is looking good. The environment, human associated. Feature, intestine environment, material, feces. Great. And now we've got some stuff. Oh, that's very helpful. Post-disease status, they all have Parkinson's. Do they truly all have Parkinson's, or do they just put that here in the SRA? I can't imagine doing a Parkinson's gut study where you don't have some non-Parkinson's patients.
Is it also because we put Parkinson as a keyword, like if we went to a study that they have controls? Is Parkinson maybe?
Yeah, so maybe. So I'm looking at, here we go. Fecal microbiota and L-delta naive Parkinson's patients. Those are all the same. The sample names are different. So if I was one of these researchers, I would happen to know that PD01 was like one patient that did or did not have whatever. So what can we do? We can go back over to this project right here, the bio project. Thanks, Adblock. Okay. So I'm just going to click on that and see if I can learn more about this study. Compare the fecal microbiomes, 31 early stage L-delta naive patients and 28 age match controls. So there are 31 actual Parkinson's patients, 28 controls. Which one is which? I don't know how to compare them here. Well, we've got accession numbers, but they don't mean anything. We've got latitude and longitude, those are all the same. And then here, it's just, this is where they should have put Parkinson or control. Instead, what we get are just patient IDs. How do we know which patient has Parkinson's and which one's in control? Maybe they don't have all the samples. So this might just be the Parkinson's ones.
When you search by project.
That's this accession here, PRJ.
Like, can you make them as hard as you can do something?
Yeah. So we can go here, and looking at SRA experiments. That's the actual sequence data. Now we can send that to the run selector. So if you follow me there, this is the whole project. Go there. Now we've got 364. Send to run selector. Beautiful. I'll download that metadata table, open it up. Yeah. The project thing. Oh, I just clicked it.
Oh.
Yeah. Since it's a hyperlink. File project. Yes. There we go. Okay, and we do see like patient number is repeated multiple times. Some are 3 times, looks like some are once, some are 4. But at least now we have who's control and who's Parkinson. So if you were gonna do this one, which samples would you pick?
It's in control, it's in Parkinson's.
We need to do 10.
5 and 5.
Yeah, okay, so 5 and 5. Let's try to make this like finishable. So, yeah, I would say 5 and 5. Get 5 controls and 5 Parkinson's, but be aware that it might be the same person. Do you want replication with the people, or do you want to pick 5 different people for each? I'm guessing there's like time points in this. So do we, where we have time point? We got collection date and they actually make it different. Ah, all the same. Is that actually possible? Maybe they took samples from everybody on the exact same day. They took multiple samples. they lock these people in a room and like make them poop 3 times.
Busy day.
Yeah,
They test in the environment, like, what is it, does that mean like, how are they getting it's like,
it's poop sample.
okay, it's.
Yeah. So yeah, here's the actual feces. So that's the material that they sample. Yeah, that's fishy to me. I think that that's not what actually happened. We could probably learn more about this study. I could try to search this accession number in Google Scholar. All right, here's one. That's not the actual paper, that's just citing it. They use this data. Oh, that's pretty cool though. Hidden associations of viruses with chronic diseases. Neat. Yeah, I'm not seeing a paper that easily links that. Go back here and look for authors, and I don't see. Sometimes they'll have an author name, so you could like track down the paper or email the person. Say, who was patient PD01? They will never respond to you. It's never happened. Tried it dozens of times. So we're a bit limited, but we've got the information that we've got. And so for this, I would just say find 5 different patients that have Parkinson's, 5 that don't. And then make a list of those accessions over here. Throw those in your accession file. And then you're off to the races. Run the pipeline.
Is there like a certain like number of bases or like fights we should try to stay like under or like say above?
Yeah, so let's look at that. We've got the number of bases, number of bytes. So that's already run table 7. So there's a number of bases versus the number of bytes. Most of them are falling in the 2 × 10⁹ range. I would not go for any of those really big samples. Or for these here. But this stuff right in here, that's about right. And so we want our bases. Yeah, let's say between 1 e⁻⁹ and 2 e⁻⁹. Okay, so back here. There we go. Bases is less than it's greater than there. We're kind of filtering down our samples to just those. Cool. So here's the samples with those. We still have quite a few. How else do we want to filter it? That's avoiding these crappy samples that kind of failed and the giant samples that are just going to be overwhelming. Slow down your pipeline delivery.
It's like the average slot meter.
That's like how long the average read is. Is it average spot length? ABG?
And then like capital S, capital M. That's literally how we those runs so often.
Okay. There. So we've got a minimum of 83. Max of 243. This could affect your QC because you might have a cutoff that like throws out any reads that are less than 100. So maybe pick samples that have an average spot length of at least 100. So we could add that to our filter. For 100. So that takes us down to 146 samples. Pick from. And then we've got the, I think it's like host disease status, yeah. That leaves us with 51 controls and 95 Parkinson's, plenty to pick from. So at this point, we're just picking 10. We don't need to be super systematic, like pick 10 of those. There, I randomly picked 5 of each. And so this would be my little table. Go ahead. So, session. There they are. You just need to be able to keep track of which ones are disease and which ones are Parkinson's somehow. So have a little table for yourself out at the end. That's it. So that's how I would like go about for this study, kind of choosing 5 each disease status. You don't have to do an R, you can just pick 10. But I think that would be a really nice project right there. It's actually got 2 conditions. We're randomly choosing 5 patients from each. We're going to run our pipeline on all 10 of those. And then at the end, our output table, we can look and see like statistical differences between gene, not expression, but gene presence in the gut. See if there's a difference between Parkinson's. What other stuff people care about other than Parkinson's? I really like that idea of changes of age. Okay, yeah, these differences. This might be trickier to find. Yeah, yeah, you have to choose like 2 ages, I guess, if you're gonna do it until like, I mean, we could. Problem is like, how do you control for that? Like, because people's guts are going to be different from each other, so you'd have to have the same person. In a time series and pick them. It's tough, and I'm not saying it's not out there. I'm just saying temper your expectations. I don't know how to sort for that in the SRA right away.
What's a longitudinal study?
It's a longitudinal study, so I might try Scholar right off the bat. Gut microbiome, longitudinal, aging. Got a systematic review. Don't want that. We want actual data. Wild animal population, is that a close enough program? That sounds good. Okay. Plant-based diets in the gut microbiome, Baltimore longitudinal study of aging. Maybe that's better. Just from last year. Okay. So now we got to find where their data. I'm betting it's here in the appendix A supplementary data. No, those are multimedia components. Here we go. No. Oh. Available on request is science speak for nobody can ever have this ever. Sorry.
Wow.
Why?
Like,
some journals still let you get away with this. I refuse to review for them. When I review articles, I do all the time. If they say data available upon request, reject, fuck off. Right? If you're not willing to share your data, it means that I can't trust your results. So, done. This one's not reproducible. That's crappy science. Nice try, people. Okay. Hmm. Maybe the wild animal one? Materials and methods. Where are we gonna find? Here we go. Sequencing. Oh, it's MySeq. They use primers. Yeah. Throw in that. Because it's not shotgun sequencing, it's just amplified radiographic CNS. So there won't be any genes in there at all. That's non-coding. Chance and healthy gene. Yes. Now, your data node. If I see available on the class, that's it.
Oh, there's one.
Um, Good job, people. Okay, so they didn't follow, and I understand why, because that'd be so hard. Like follow the same people over time. That would take 20 years for you to like get results. So they're grouping people into age groups, and kind of looking at generalized trends. Yeah, that's cool. It'll complicate things for us if we're just picking like 3 from this group, 3 from that, 3 from the other. Um, but our actual results don't matter. It's just an intro class. It's fine. So yeah, this could be a good one. So we go to our SRA experiments, send them all to the fund selector, download that metadata. And so let's see what we've got here. Tell who's in what group. No. Maybe these have a clue.
They're like,
Young, medium, and large, late. I would go with that since they're not providing any like column with the actual stuff. A lot of times they get encoded in sample names like this, so I would pick, a few from each of those. M, Y, and L. And let's just assume that it actually means young, medium, and late, or something like that. I don't know.
Yeah, there are all these different groups of being old. You have young old, middle old, and long lived old.
Right. I mean, it's probably a safe assumption. If I was doing this for publication, I'd probably contact those authors, find a friend who speaks Mandarin and send them an email, right? And say, can we safely assume that L means large or whatever? So yeah.
you said the basis of the smalls is like.
Yeah, they use a technology called NextSeq, which puts out a crap ton of data. So these would take longer to run for assembly, because it's just a whole lot more data than the other study, but it'd still run. So remember, like, for the longest, by far, the only, like, our bottleneck in terms of time is gonna be that assembly step. And that one we're submitting as 10 separate jobs. And so they all kind of run at more or less the same time. So it's okay. I'm hoping that not many other people here on campus are gonna be running a bunch of jobs on Bora over Thanksgiving. Even if they are, they can't take more than a day or two, and you're gonna get finished up by the end of the week, no problem. Okay, so yeah, that's a pretty cool study. PRJNA 1195999. You want to look for some more? There, anyone care about non-humans?
Was there something you would do for research that we could look into? We could put on there? Or-
I'm gonna keep you away from my research for your own good. My problem is that I don't care about bacteria. I care about fungi usually, and that's a whole different problem for annotation. Because eukaryotic genes have introns, alternative splicing. Bacteria are just simple, easy biological machines that follow the rules. So we're using Proca, it only works for bacterial and archaeal.
It's about direct manufacturers, that's what.
Okay.
Seems interesting. And we say that's. I don't
Like
know,
shotgun.
yeah, but I don't know why where they keep it.
Good question. What's like my scholar search to get to that, the title?
Stress related to interest in the government.
So we're talking traumatic injury. Okay. Let's scroll down. Okay, we've got our sequencing. No, 16S. But let's see, usually it's at the bottom. There's like a data availability statement that'll show up. Like, searching for PRJ, I see nothing. Don't see the word available. You know? So they haven't even made a statement that I can tell. They're pretending. It's a real journal. I had to know. Oh, well, any of it.
Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find something about it. Yeah, I was trying to find
maybe include like soil bacteria, like maybe. Like
I
different.
mean, you don't have to do soil just because I'm sitting here. It's cool.
I'm trying to think of something that isn't like, that has the. So.
I'm gonna get something where I want to like things.
Okay.
Places.
But.
Resilience and methane cycle and microbial function genes to drought and flood in an alkaline wetland. Metagenomic study that looks promising. Rapid reshaping of the soil microbiome and metabolome of short-term flooding and draining in rice. This is like an ag thing. Primers.
So.
16S. Dang it. And don't get me wrong, 16S is really, really nice. If you want to know who's there, it's the way to go.
Yeah, this paper, I kind of want to see that talking about like mercury methylation activity. And here they did 16S amplification, but they also did assembled measurement data. So they have data in house at 2 different places.
Yeah.
Yeah.
This is not uncommon. Because it's really pretty cheap to do 16S or ITS sequencing. And it will answer your questions about who is there and what relative abundance. And it's a lot more expensive to do the shotgun stuff. And so people will like do a 16S study and then pick just a few key samples and do shotgun on those to kind of like get an overview of what's going on. So if it's housed in 2 different places, that's fine, but do they give like a PRJNA number or whatever?
Let me go back there. Yeah, they did for the 16S stuff for the metagenomic data. It sounds like this thing that I'm not familiar with.
Okay.
Yeah. Available at img/m
Oh.
gold study
Okay.
ID.
So to search for that, it's JGI-IMG. That's the Joint Genome Institute,
Oh,
Integrated Microbial Genomes and Microbiomes. So, oh, they changed the website. Okay, and so this is just another repository. It's also backed up on Sequence Read Archive, but you have to kind of know how to make them match each other. So do they give like a number or anything?
yeah, capital G lowercase s 0140997.
Cool. So we got 12 samples there.
Oh.
Nice. So there's the gold study ID. Mercury Terabyte project. Neat. So this is a bit different from the SRA. So it'll be tougher because our pipeline takes an SRA session. So we would, in order to use our pipeline, we'd have to find the SRA accession that match up to these. Which isn't impossible, but it's also not straightforward. See it right off. I'm gonna say avoid for now. JGI, you have to like make a user account and find stuff to download. Are two different projects enough? Give me a third. Yeah. If you decide later on solemn reflection that you don't like them for these and want to do a third and form a third group, great. But otherwise, it looks like we got a couple decent samples. We got the young, middle, and late people, and then we've got some Parkinson's and non-Parkinson's. So I would want you to team up on those as you see fit. Whichever set of samples you feel like working on. Don't wait for me to assign you. I'm not going.
Could you put like the search strings in that one like lesson?
Yeah. So this is the project ID for young, medium, late microbiomes. Oh yeah, sorry. It's for the Parkinson's. And then yeah. There we go. And that's for the aging. Okay. What I'm envisioning is we'll have two teams. One team picking the aging, one team picking Parkinson's. Everybody on that team will be responsible for running the same 10 samples as everyone else on the team. So as a group, you decide which samples you want. And then everybody will be running almost the exact same pipeline. What will you change? That's between you and your group also. What steps in this do you think would be interesting that you think might make a difference? Quality control could make a big difference downstream. The way that things are assembled can make a big difference. The way that things are mapped back onto your assemblies can make a big difference. Right now, our pipeline is basically just using defaults all the way through. Bring it up. Next. So setup and download, those aren't going to change. All that's going to change is you're going to be downloading from a new list of sessions. We've got the quality control. There's the fast P Oh, that one's gonna have to be turned into a for loop. Yeah, I don't know if you guys have that. That shouldn't be too troublesome. Especially because we did that in the last assignment. So what am I doing here? This was the only real thing. N0, L100, E20. That's the whole quality filtration. So that could change up. If you change the minimum length from 100 to 200, depending on your samples, you may get no reads out the other side because some of these are only 150 long. So think about what you might change there for your quality control. You could allow some ends to be present, those uncalled bases. So you can make this a bit sloppier. Or you could change the length cutoff. You change the expected errors. Then we've got the assembly. That should default everything. Except I told it use 20 threads. Didn't want to take forever. But Spades gives us a lot of choices. So we could try only assembler. That gets rid of the error correction step. You could try the careful flag. Where
Doesn't matter what order you put the flags in.
you have to keep the meta flag. Manage on it. Yeah. Oh yeah. The Kamer sizes. Must be odd and less than 128. So that looks like that's the default. Yeah. 27 or maybe it's 21. Yeah. 21, 33, 55. You say 21, 55, 77.
But this changing anything since.
It uses more memory, but it's because those Kamers are every possible word that is that long. So it runs through all your reads saying like, okay, every possible 21 letter word that's in here. And then it does it with 55 and then by default it does 33 and then 55. So it's trying 3 different lengths of words and it's combining them all at the end. The longer your words, the more specific it's going to be. And so you can get better assemblies quite often. So 21, 33, 55 is the default, and that gives you usually pretty good assemblies. If you throw in something like 127, your assemblies will be better out at the end. So they'll be longer. There'll be fewer of them. Why don't people do this every time? You might run out of memory. And also, all of our reads aren't even 127 long. So that's actually impossible with these short reads. We had a cutoff threshold in our quality control of 100. So our camera size can't be more than 100. We don't necessarily have all of our reads that long. You can do 99 there. Unless you change the QC and say cutoff of 150.
So does that mean like all this like short, like when they consider it like a maximum of 100 or like
Minimum.
minimum of 100?
Yeah.
Why do you need to, like, if we're gonna go to 99, just do 99, if you need like it?
Yeah, okay, so they. That graph theory stuff, okay. But essentially, we do it 3 different ways. The most permissive, like 21, gives us a framework to work from. Then we go from the 55s, and only where it makes changes to that De Bruyne graph will it actually do it. And so it's like improving iteratively your assembly. Yeah. If you go straight to 99, you actually don't get as good of a graph. It's nice to improve it. Yeah.
And then going back to the pieces like the length, so the minimum of 100 will like, throw out like the really short ones that kind of like build over. So you want to like increase it to like be more accurate, but like in like decreasing it would be like you get
You
like,
get,
first
yeah,
phase.
so right here,
Mhm.
this L flag is the minimum length. And so this is throwing out any read from your files that isn't at least 100 long. So the higher this number, the more stringent you're being, and you're throwing out more data. Now you may want that. You may say like, I don't care about losing data. I really want the highest quality possible stuff. I'm going to get rid of anything that's shorter than whatever. But that goes back to your like average slot length,
Mhm.
right? So we look here from this study, the max was 243. Median of 177. Okay, so I would not want to set my QC minimum length to anything less than 177 here, or I'm throwing out half my data right off the bat.
So not less than 117.
For this data set,
Yeah.
right? So what I'm saying is that for that data set that we're looking at here, which I believe is the Parkinson's people, if I set my quality control L to 250, I wouldn't have any data left. So this number makes it more and more stringent to a point, and then suddenly it kills your entire pipeline. So what's the minimum length of reads that you want left over after quality control? We just went with 100 for no reason necessarily. 150 might be great. Be throwing out a fair amount of data. Here's the read lengths. Just a quick look. Okay, so we're really only throwing out a little bit if we picked 150.
So you would say like 150 would be the lowest you've heard.
For this, there would be no, for these particular samples that I randomly chose from this study, 100 and 150 would make no difference because there's nothing between there. So we've got some really low reads. Those are going to get tossed out whether I pick a cutoff of 100 or 150. If I pick a cutoff of 200, then I'm throwing out a whole lot of data. And maybe that's what I want. Maybe it's worth trying for one of your pipelines that you decide to run. Like, okay. Let's see what happens to our conclusion if we do make the cutoff 200. Yeah, we're going to lose a lot of data, but the stuff that comes out at the end is going to be higher quality. Maybe that'll give us a better assembly overall. Maybe that changes some of our answers. What you can't do is say 300 here. Then you lose all your samples.
So a good healthy rate would be like 100. Just based on like looking at this.
Yeah,
So
just looking at this, I'd say you can cut off below all these samples or above them. That'd be like a substantial change to the pipeline. So that's the QC, that's the assembly, and then there's annotation. What do we do here? Basically just the defaults. I told it to use 20 CPUs. Kingdom bacteria, we're not going to get away from that. It is a metagenome. Let's see. Okay. Beginner, moderate, specialist, expert, wizard. So we're seeing we can do some different stuff. So we could set a value, for example. That's the expected errors, or like how well things going to match up. The smaller that number, the more stringent. So what is ProPed doing? It reads your contigs, your big long sequences that you built. It's looking for start and stop codons that look like a gene. And then it's taking that region and mapping it up against its database of known bacterial stuff. Whether it decides that it looks like a specific gene in the database or not, based on that e-value. So the smaller the e-value, the more picky it's going to be about saying, yes, this is definitely a gene. But it's definitely this gene. So you could raise that all the way up to 1, and you would get all kinds of hits. You'd get much lower proportion of hypothetical proteins out of the end. But do you trust them? Trust them less, at least.
Can you change the database or something too, like the diamond database or the last database?
Good man.
Is there like a better one on like the cluster that we can use for this, or should we just use the standard one?
The standard ones are pretty good. If you feel like it, you can make your own database of anything. Okay, so if somebody wants to take that on, fine, but you're adding another script to your pipeline. Here's a crazy person. All right, so you can change a lot of stuff about how this thing runs. Really tiny e-value. We're giving it just a list of valid plasmids that we know, like from our previous work in this lab, and we're saying just map it to these. Use the genus level database. Here's the genus code that I'm looking for. So this is a person just looking for E. coli plasmids in their metagenome for a particular strain, particular plasmid. Okay, so they've got a very specific question, and they're asking ProPed to find stuff that way. Got a whole bunch of different things that we can do. You could just change the RFAM. Search for non-coding RNAs within the internal RFAM. It says, warning, slow. So maybe for a project over Thanksgiving, don't pick that. That's one of your things. All right. The bigger picture here is that I don't care what you change. What you need to get into your group before we leave today in the next like few minutes. And exchange numbers, whatever it takes, and then pick your samples. Or make a plan with your group to pick the samples and decide who's going to do what differently.
And you want each person to change one parameter.
There, doing change a bunch. Now, here's the thing. All of you are gonna be running the same pipeline, just with slightly different tweaks in some of these parameters. You're gonna get the same file names out at the end. That's bad. So you're gonna have to work out with your team, how am I gonna rename my files? Whether it's your initials, you put those in the initials or whatever. I like naming files with the extra parameters. So.
Is that something you just like to the end, you know, to get like them switch up into the left hand?
You could just do it at the end, yeah. But what's going to be impossible is if you have 4 different versions of the same pipeline with the same files, which one came from which, right? So then I could go and do the stats on this and say, great, there was a difference. I don't know which one was different. And that's weird.
How do I use?
I don't know yet.
Okay.
Yeah. Your output files aren't going to be too big, so maybe we can just like get up. Really, it's just that one little output file that's like, here's the genes and how abundant they were. It's a tab separated thing. All right, we got a few minutes. Find your groups, exchange numbers.
Oh.
I can also get practice.
Okay.
We're divided.
Okay,
Yes, sir.
that was easy. All right, so we got aging and we've got Parkinson's. Okay, we happy with this? Okay. Choose your samples wisely. Make sure you all pick the same accession numbers. Yeah. Cool. Okay, so so logistics. For the rest of the semester, this is open work time. And I can help you with questions, with bugs, with whatever. It's just gonna be, we're here working on this together. Yeah. It does not have to be finished by the end of Thanksgiving. Okay. I want you to have a nice break. All of you little babies are so stressed out and you got finals and stuff coming up. It's fine. I also don't want to do anything right. I also have the professor equivalent of finals and papers due. And I don't want to think about them for a little bit. I just got a new PlayStation game. Ghost of Yote. It's so good, but I only got to play it for like 2 hours. So yeah, I'm gonna have a full 24 hours of just that. So yeah, take care of your brains, have a Thanksgiving break. Yes, jobs will be running over Thanksgiving, and that's good because that's when you'll get your errors and whatever. The rest of the semester here, come into class, we're talking through it, we're working through your pipelines, and we're just gonna be like colleagues on the bioinformatics team trying to make this work. There is no final exam in this class. Okay.
Script 3, did you get an out of memory error?
Thank you.
That's bad. I ran it like over them. Which one were we supposed to run last time? The 4.
Yeah, I think we did.
Okay, so you got out of memory, submit it to Kuro. If you get out of memory, submit to Kuro, it has 384 gigs. In fact, this is a good thing to bring up. Your assemblies, maybe just submit those to Kuro to start with. So they can get real memory, that's it. Submit them to Kuro.
Okay.
Yeah.
It's skip.
Yeah, for your assemblies, and all you gotta do is log into Kuro and submit your Storm script and it will go to Kuro there.
Gotcha. Okay.
All your files, it's everything's the same, it's just when you do SBatch, it goes to the high memory stuff.
Gotcha. Thank you.
I thought.
Did it,
I don't know.
did you check the
I
output?
would have not been like that. Oh, really? Okay. Types of.
Yeah.
Everything.
Okay, so Sarah and I were working on this morning, and I think we found a problem. It was changing the names of the FASTA headers during the annotation stuff. And so then when it maps, it didn't match to anything because it looks for that name. So
I see.
today, starting here in 10 minutes, I'm gonna like write a script that fixes that, put it in, just like rerun step 5.
Okay, I need to change step 4, I add like changes like
Yeah,
So this is like, and then.
that's the thing.
Yeah, okay.
Did you add the like center
in the compliance or something.
Yeah, the compliance center, that's what screwed up.
There's
Oh, I
a-
didn't say it. That's what screwed
I
up
think
the
there's some-
mapping later,
Yeah.
that
I like.
that changed the names, so now it can't map by
You
name.
just like go to
Because
the.
I did it with that, it took like 40 seconds or like, it's like, it's not.
Yeah, I found nothing.
Okay.
I'll get that fixed today, I hope.
So our SRA one table has 3 like different information, so how are we going to get clean out of that?
You can say 3, 2, 4, and it's just like, see like 4 in the middle group.
Yeah, same time.
Or
But also like that.
Well, I gotta go upstairs, you know, what I'm saying, so you're doing. Yeah.
Have fun, everybody. I'll be around to help somewhat during Thanksgiving. But might be like a bit slow to turn an email. I got a policy that like, I'm not gonna email students when I'm even slightly drunk, and so.
It's a
So, yeah,
I'm learning my own life, I feel like I'm using this lab.